Krebs cycle, citric acid cycle. Various steps of these reactions are as follows. In the first reaction, one molecule of acetyl coenzyme A combines with 4-carbon oxaloacetic acid. With the result, 6-carbon citric acid is produced and coenzyme A is released. This reaction is catalyzed by enzyme citrate synthase. Acetyl coenzyme A reacts with oxaloacetic acid. In presence of citrate synthase enzyme produce citric acid and coenzyme A. Now, citric acid is isomerized to isocitrate, isocitric acid. Cis-aconitic acid is converted into isocitric acid with the addition of water in the presence of iron-containing enzyme aconitase. Oxalosuccinic acid in presence of oxalosuccinic decarboxylase produces alpha-ketoglutaric acid and carbon dioxide. Alpha-ketoglutaric acid reacts with coenzyme A and NAD in presence of alpha-ketoglutaric acid dehydrogenase to produce succinyl coenzyme A, NADH, hydrogen ion and carbon dioxide. Succinyl coenzyme A reacts with water, GDP and inorganic phosphate in presence of succinic thiokinase to produce succinic acid, coenzyme A and GTP. GTP reacts with ADP to produce GDP and ATP substrate phosphorylation. Succinic acid reacts with FAD in presence of succinic dehydrogenase to produce fumaric acid and FADH2. During Krebs cycle, 3 molecules of NAD positive and 1 molecule of FAD are reduced to produce NADH and FADH2 respectively. These are linked with electron transport system ETS and produce ATP by oxidative phosphorylation. This may be summarized as pyruvic acid reacts with 4 NAD positive ions, FAD positive ions, 2 molecules of water, ADP and inorganic phosphate in the mitochondrial matrix to produce 3 molecules of carbon dioxide, 4 molecules of NADH, 4 hydrogen ions, FADH2 and ATP. In the end of Krebs cycle, glucose molecule is completely oxidized. From one glucose molecule, 2 pyruvic acid molecules are formed. After oxidation of 1 pyruvic acid molecule, 3 carbon dioxide molecules are released. Thus, in all 6 molecules of carbon dioxide are released. Electron transport system. By the end of Krebs cycle, glucose molecule is oxidized completely, but the energy does not release till NADH and FADH2 oxidize through electron transport system. The metabolic pathway through which electron passes from one carrier to another is called electron transport system or electron transport chain or mitochondrial respiratory chain. It consists of a series of coenzymes and cytochromes that take part in passage of electrons from a chemical to its ultimate acceptor. The passage of electrons from one enzyme or cytochrome to the next takes place with a loss of energy at each step. It is operative in the inner mitochondrial membrane. The electron carriers include flavins, iron-sulfur complexes, quinones, and cytochromes. Most of them are prosthetic groups of proteins. Electron transport system in mitochondria consists of 4 complexes which are found in bases of stocked particles in the inner mitochondrial membrane, and also ubiquinone or coenzyme Q and cytochrome C, which are not bound to stocked particles but act as mobile electron carriers between the complexes. Complex 1 consists of NADH dehydrogenase or NADH2 reductase, which contains a flavoprotein FMN, flavin mononucleotide, and is associated with iron sulfur proteins. This complex is responsible for passing electrons, also protons, from mitochondrial NADH to UQ, located within inner mitochondrial membrane. Complex 2 consists of succinate dehydrogenase, which contains a flavoprotein FAD, flavin adenine dinucleotide, in its prosthetic group and is associated with non-heme iron sulfur proteins. This complex receives electrons, also protons, from succinic acid, which is oxidized in Krebs cycle to form fumaric acid, and passes them to ubiquinone UQ. UQ also receives reducing equivalents via FADH2 that are generated during oxidation of succinate through the activity of energy succinate dehydrogenase in Krebs cycle. Complex 3 consists of ubiquinol, cytochrome C and cytochrome BC1. The reduced ubiquinone is called ubiquinol. Here, ubiquinol is oxidized with the transfer of electrons to cytochrome C via cytochrome BC1. Cytochrome C is a small protein attached to outer surface of the inner mitochondrial membrane and acts as a mobile carrier for transfer of electrons between complex 3 and complex 4. Complex 3. This complex is called QH2 cytochrome C reductase complex. This bears three components, that is cytochrome B, non-heme iron sulfur FES, and cytochrome C1. Coenzyme Q is also involved between FES and cytochrome C1. Now, cytochrome C1 transfers electrons to CYC. Like coenzyme Q, CYC is also mobile carrier of electrons. Complex 4 is known as cytochrome C oxidase complex. This contains cytochromes A and A3 along with two copper centers. This complex receives electrons from cytochrome C and passes them to 1/2 O2. Two protons are needed and H2O molecule is formed. Terminal oxidation. Here, O2 is ultimate acceptor of electrons. It combines with protons to form metabolic water or respiratory water. Complex 5. When electrons are transferred from one carrier to next carrier via complexes 1 to 4 in electron transport system, they are coupled to ATP synthase enzyme complex, complex 5, for production of ATP from ADP and inorganic phosphate IP. Here, number of ATP molecules synthesized during ETS depends on nature of electron donor. Oxidation of one molecule of NADH gives rise to 3 molecules of ATP and 1 molecule of FADH2 gives rise to 2 molecules of ATP. ATP synthase complex is called complex 5. Complex 5. During transportation of electrons, hydrogen atoms split into protons and electrons. The electrons are carried by cytochromes. Before last stage, where hydrogen atom is accepted by oxygen to form water, the electrons again recombine with their protons. Oxygen acts as final hydrogen acceptor. Oxidative phosphorylation. The whole process where oxygen effectively allows the production of ATP by phosphorylation of ADP, is called oxidative phosphorylation. In other words, synthesis of ATP is called phosphorylation, and as it takes place in presence of oxygen, it is called oxidative phosphorylation. The enzyme required for synthesis of ATP is called ATP synthase. This is located in F1 or headpiece of F0 to F1 or elementary particles. ATP synthase enzyme becomes active in ATP formation where there is a proton gradient shading higher concentration of H2. Net gain of ATP. Complete oxidation of glucose to CO2 and water shows that there is a net gain of 38 ATP. Each NADH plus hydrogen ion produces 3 ATP molecules, while FADH2 forms only 2 ATP molecules at the end of reaction. Thus, total gain of ATP in aerobic respiration is as follows. Glycolysis produces 8 ATP. Pyruvic acid produces acetyl CoA, which produces 6 ATP. Krebs cycle produces 24 ATP. Total 38 ATP. However, in most eukaryotic cells, 2 molecules of ATP are required for transport of NADH produced in glycolysis into mitochondrion for further oxidation, and therefore net gain of ATP is 36 molecules. Significance of Krebs cycle. During Krebs cycle, carbon skeletons are obtained for use in growth and maintenance of the cell. Many intermediate compounds formed are used in synthesis of other biomolecules such as amino acids, nucleotides, chlorophyll, cytochromes, and fats. In Krebs cycle, amino acids are synthesized from alpha-ketoglutarate acid, pyruvic acid, and oxaloacetic acid. It is major pathway for generation of ATP molecules, which make energy currency of the cell. Energy released from glucose is used in various biochemical reactions. Krebs cycle is directly related to nitrogen metabolism. Alpha-ketoglutarate acid and intermediate is the first acceptor molecule of NH3, forming an amino acid, the glutamic acid. From glutamic acid, various transamination reactions begin to form different amino acids, which ultimately condense to form proteins. Krebs cycle is also related with fat metabolism. After beta oxidation, fatty acids give rise to active 2C units, the acetyl CoA, which enter the Krebs cycle.